Institute for Stem Cell Science and Regenerative Medicine is an autonomous institute of the Department of Biotechnology, Govt of India. DBT-inStem's mandate is to carry out research in stem cells, regeneration and repair for understanding development and disease, but also with an emphasis on clinical translation. To focus and build capacity in these areas, our research is collaborative and interdisciplinary in nature, as we aim to do research that goes beyond the scope of a single laboratory. DBT-inStem shares a research campus and facilities with the Bangalore Life Sciences Cluster, which includes the National Center for Biological Sciences, the Center for Cellular and Molecular Platforms, and the Tata Institute for Genetics and Society's Center at inStem. This unique transdisciplinary environment facilitates collaboration across various disciplines and helps take innovative ideas for technology development, as well as to use the knowledge gained from basic stem cell research to clinical application. inStem harbors extensive expertise in stem cell biology and regenerative science. The stem cell facility at inStem can provide expertise and information for generation of induced pluripotent stem cell lines from various human cell types, as well as differentiation and quality control of these. We have the capability for an extensive biorepository that has been set up, and also expertise in generating various kinds of human stem cell derived organoids such as cardiac, cerebral and lung organoids and various skin epithelia.
My lab studies the mechanistic basis of neurodevelopmental disorders. We use the stem cell facility because we need a very sterile environment to do these experiments. This is very important because this maintains the sterility of the entire area and prevents the entry of microbes, dust, and this is a very important requirement when we are culturing stem cells, which are sensitive cells which cannot be grown with the help of antibiotics.
We are working on a group of heterogeneous heart muscle disease called cardiomyopathies. For the same purpose, we use stem cell core facility to generate iPS lines from the patients, and from these induced pluripotent stem cells, we actually generate the cardiomyocytes, and we use cardiomyocyte to study the pathogenesis of the disease and drug screening for this particular disease.
We in the lab are interested in understanding the role of tubulin and its post-translation modifications in regulating functions of cilia and flagella, and how the defects lead to human disorders called ciliopathies. Now for this, using the stem cell facility at inStem is extremely beneficial because this allows us to generate organoids of organs like kidney, trachea, lungs, the retina and so on, which allows us to understand the molecular mechanisms leading to ciliopathies in a dish.
My laboratory studies the lower urinary tract, which is comprised of the bladder and urethra. For my work, the stem cell facility is very useful as it allows me to model the epithelial barriers of the bladder and urethra in a 3D state. This allows me to study both epithelial differentiation and study infection in a dish.
The stem cell facility also offers training and consultation in gene editing approaches, as well as in setting up screening platforms for high throughput screening using various stem cells.
Also, the stem cell facility aims to generate a comprehensive array of fluorescently labeled reporter human pluripotent stem cell lines by CRISPR-Cas9 mediated genome editing. This helps us in developing an important toolbox of various reagents to understand the cell's ultrastructure during pluripotency and differentiation. These tools will be helpful to develop high-throughput, fully automated genetic and small molecule screening platforms. To know more about us, please contact us through our website.